Oh, yeah.
Now you can start.
So, hello everyone. Myself Kiran Tak, and today's topic of the discussion is chapter 11, that is biotechnology principles and processes, which is taken from class 12th biology NCERT. So, let's begin the session with the understanding of the term biotechnology. Biotechnology involve the techniques that uses the organism itself, or we can say the enzymes from the organism, so that various products can be produced for humankind. And there is an other definition that is given by the European Federation of Biotechnology, that is the integration of natural science and organisms, cell parts thereof, and molecular analogues for products and services. It is a modified definition that again explains us the involvement of natural science and organisms, cell, with the molecular technique that can be useful for the production of the desired products. Now, the biotechnology involves various microbe mediated processes, like for the making of curd, bread, or wine. But in restricted sense, the all the processes that are involved in the genetically modified organisms are comes under the biotechnology, like in vitro fertilization that lead to the formation of the test tube baby, synthesizing a gene and using it, developing a DNA vaccine, or correcting a defective gene, and various other fields where biotechnology is used, that are animal breeding agriculture, bioinformatics, proteomics and genomics, plants agriculture or crop improvements, recombinant DNA technology, healthcare or pharmaceuticals, medical diagnosis, fermentation technology, improvement in food technology, environmental and energy management, and etc. Now, the whole chapter is divided into three different phases. First is the principles of biotechnology, then tools of recombinant DNA technology, and the last is the processes of recombinant DNA technology. So, now begin with the principles for biotechnology. So, in this chapter, the principles of biotechnology are divided into two parts. First is the genetic engineering, and the second is bioprocess engineering. So, the genetic engineering, it is the technique that alters the chemistry of genetic material and then introduce it into the host organism, where it the phenotype of the host organism change. And this technology is called also called as recombinant DNA technology, in which the desired trait is generated. And there are advantages of sexual reproduction over a sexual reproduction, and the major one is the variation, and the other is the new formed individual has characteristics of both the parents. Okay, so variation occurs due to the recombination uh, uh, takes place when the uh, two gametes are fused, and due to which this the new organism formed have both the characteristics of the parents. So, by taking the advantages of the sexual reproduction, the traditional method is used to produce required product. This traditional method is also known as traditional hybridization, or we can say desired cross. So, according to the name, it suggests that the when there are two different organism and there are two different phenotype that are present in these organism, so we will uh, cross these two organisms having two different desired phenotype, and the newly formed offspring after the cross having both the characters from the parents. So here we can see that this is the donor variety DNA strand. It has the desired gene, This is the desired gene, and this is the recipient variety of the DNA strand. So when there is a cross done between these two gene, and then the new variety of the DNA strand will form. But in this case, with the desired gene, so with the desired gene, this is the desired gene, undesired genes are also present. Okay, and sometimes we didn't even get the desired gene. So in modern technique, what we have done is, first we isolate our desired gene, and using various techniques like gene cloning and gene transfer, we overcome the limitation of traditional hybridization. Then we introduce this gene into the host organism, where it can clone and give the desired product. So as we can see in the given figure, that this is the insulin producing cells. Okay, and first the whole genome is isolated. This is the whole genome, and then our desired gene of insulin is cut with the help of the enzyme. This is the desired gene, and then this desired gene is uh, 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 ligated with the other DNA. So this is the bacterium which has the plasmid. Plasmid is removed from this bacteria, and then this plasmid is fused with the gene of interest, and then it is incorporated into the host organism, where it is cloned and then expressed. So we can get our desired product. Now, the next is the bioprocess engineering. So, the bioprocess engineering involves various engineering processes to generate the desired microbe or eukaryotic cell in large quantity to produce various products like antibiotics, vaccine, enzyme, as it produce sterile condition and optimum condition to the desired product, and hence we get higher yield of the required product using this bioprocess engineering. So now we will discuss the genetic engineering process in detail. So the first is the construction of an artificial recombinant DNA molecule. So first, Stanley Cohen and Herbert Boyer in 1972 construct an artificial recombinant DNA. In this uh, in this construction, what they have done is they used salmonella typhimurium from the plasmid, the native plasmid. The antibiotic resistance gene is removed with the help of the molecular scissors, that is known as restriction enzymes. Okay, so this is the foreign DNA, the whole genome of the Salmonella typhimurium, and this is the vector. And this is the gene of interest. This is the antibiotic resistance gene, which is ligated with the help of the DNA ligase enzyme, and then it will form the recombinant DNA molecule. This recombinant DNA molecule is then transformed in the E. coli, and then the with the cloning, with the help of the replication process, the cloning of the gene will takes place. So here we use the vector DNA plasmid. So vector is used to transfer the piece of DNA that is attached to it. As in malaria disease, mosquito is used as a vector, so that the parasites is transferred into the human. Likewise, the gene of interest is incorporated into the host organism for cloning purpose with the help of the vector. So vector play a very important role here. Now, as we have seen that here the host organism is E. coli, where the multiple copies of the antibiotic resistance gene will be formed, and this process is called as cloning. So, the recombinant DNA technology involves following main steps. First, the selection of the desired gene, selection of vector for the transfer of the gene, that is known as cloning vector. Here, plasmid is the cloning vector. Insertion of recombinant DNA into the host, then maintaining the introduced DNA, maintaining the recombinant DNA into the host organism so that it can pass to the next generation. Now, after completing both the principles of the biotechnology, now we will move forward to the tools of recombinant DNA technology. So, as in any type of engineering, there are some tools that are required for the engineering. So, in the recombinant DNA technology, the various tools are restriction enzymes, DNA modifying enzymes, cloning vectors, and competent host that is used for transformation with the recombinant DNA. So, uh, the idea of using the first is the restriction enzymes, and the idea of using the restriction enzymes came from the defense system of the bacteria. So in 1963 it was found that when bacteriophage, this is the bacteriophage and this is the bacteria, so the virus that infect the bacteria are known as the bacteriophage. So bacteria infect the host cell, it transfer the genetic material inside the bacteria. The bacterial defense system activates and consider the genetic material of the phage DNA as the foreign DNA. Thus, it produces the restriction endonucleases that acts on the phage DNA and cleave it into the small non-infectious fragment. And the restriction endonucleases are the enzymes that have two major rules. The first is the addition of the methyl group, and the second is the cutting of the DNA. So the methylation is necessary so that it cannot cleave its own DNA and cut it will cut the DNA of the foreign cut the foreign DNA. So here hind 3 is the in this figure, hind 3 is the foreign DNA, and it cleaves the uh, DNA into two parts and make the fragments. Now the first restriction endonuclease was hind 3 in 3, but it was characterized after 5 years. The hind 3 hind 2 sorry hint to always cut the DNA molecule in a particular point, that is the recognize the specific sequence of the 6 base pair, that is known as the recognition sequence or recognition site. There are more than 900 restriction enzymes that have been isolated from over 230 strains of bacteria, and each of the restriction enzyme recognize different recognition sequence. So, how the nomenclature of these restriction enzymes has been done? So, let's take an example of the eco R1. So, eco R1 here, the first letter represent the name of the genus. In this case, the genus is Escherichia. CO is for the species. It is say coli. R represent the name of the strain. In this case, the strain is RY13, and the Roman number 1, it indicate the order in which the enzymes were isolated from that strain of bacteria. Okay. So the restriction enzymes belong to a larger group of the enzymes that are known as nucleases, and the restriction enzymes are of two types. First is exonuclease and the other are endonuclease. So exonucleases remove the remove nucleotide from the end of the DNA, whereas endonucleases make cuts at specific position within the DNA and make small fragments. Restriction endonucleases inspect the length, it recognize the specific sequence and then cut it into the small fragments. What it does is it break the sugar phosphate backbone between sugar between two sugars. It actually recognizes a specific sequence, that is palindromic sequences. So what are palindromes? So in simple way, palindromes are group of letters that form the word, as we can say that we can see in the Malayalam. So if we read Malayalam in the forward direction, or we read in the backward direction, it is the same word Malayalam. So it is again, in the case of the DNA, if we read, if like eco r1 recognize GAATTC sequence, then if we read the sequence in the 5 prime to 3 prime direction, in both the cases, in template and its complementary sequence, in 5 prime to 3 prime direction, it is GAATTC only. So it is true if it is read in 3 prime to 5 direction also, like CTTAAC in both the cases. Now, uh, the restriction enzyme can cleave the DNA fragment in the sticky and form sticky ends or blunt end. So these are the sticky ends. These both are the sticky ends. So now, the enzyme cut both the strand at the same site. So this red one is the vector DNA, and this black one is the foreign DNA. So now we will understand that how these sticky ends are formed and how they are joined with each other. So the vector, if the restriction enzyme we use is eco r1, so it recognizes GAATTC, so it will cut between G and A here, and in the complementary sequence here, in case of the vector DNA. And in foreign DNA, again, if this sequence is present, then again it will cut here and here. Now, after the cutting, sticky ends are produced, and with the help of the enzyme ligase, these sticky ends join with each other, and the recombinant DNA is formed. That means this insert is now incorporated in the vector. The next is the blunt end. So the blunt end are also known as non-cohesive ends, and they, in this case, both strands terminate in a base pair, and they are joined with the help of the enzyme ligase. These are some of the example of the uh, restriction endonucleases, like EcoR1, HINT3, BAMH1, SAL1, BAL1, HAE1, SMA1. And these are sources of these enzymes. These are all the recognition site, and from where it will cleave it, and what type of cut they will form, like sticky or blunt. Now, the type of restriction endonucleases. So there are three major types of restriction endonucleases: type 1, type 2, and type 3. So the type 1 enzymes are bifunctional enzymes and having three subunits. They recognize The recognition site is bipartite and asymmetric. Bipartite means the recognition sequence is recognized by two subunits. Here the restriction and methylations are naturally exclusive, and the cleavage site is non-specific, and it is more than 1,000 base pair away from the recognition site. That means it will cleave more than 1,000 base pair away from the recognition site. Next is type 2. So type 2 restriction endonucleases have separate endonuclease and methylase uh, uh, methylase activity, and the recognition site is 4 to 6 base pair sequence. It is often palindromic, and the restriction and methylation uh, reactions are separate reaction, and the cleavage site is same or close to recognition site. And the next is the type 3, which is bifunctional enzyme having two subunit. It has 5 to 7 base pair asymmetric sequence. The restriction and methylation takes place simultaneously, that means one by one, and the cleavage site is 24 to 26 base pair downstream of recognition site. As we can see here that the in case of type 2 restriction endonucleases, the cleavage site is same or close to a recognition site, so it is very helpful for the genetic engineering process. And now and uh, type 2 restriction endonucleases are only used in this technique. Now the DNA modifying enzyme. So as we are playing with the DNA, but there are so many changes that are required during the genetic engineering process, and this can be done with the help of the enzyme only. So the first is DNA polymerase. So the function of polymerase is to synthesize the copy of nucleic acid by elongation of the uh, by forming a new DNA from the template DNA. The next is the reverse transcriptase. So it is an enzyme that is used to generate a complementary DNA from a RNA template, and the process is known as reverse transcription process. The next is alkaline phosphatase. So there are two alkaline phosphatase that can be used: bacterial alkaline phosphatase and the calf intestinal alkaline phosphatase. They remove the phosphate group from the 5 prime end of the DNA, and they are used to prevent the unwanted ligation of the DNA molecule that can create a problem during the cloning, because sometimes the vector get itself uh, ligated without the gene of interest, so it will not form the recombinant DNA, and it will be problematic for the cloning process. The next is the polynucleotide kinase. So it catalyzes the transfer of the terminal phosphate, that is the gamma phosphate, to the 5 prime hydroxyl termini of the polynucleotides such as DNA and RNA, and also in oligonucleotides or mononucleotides. It is used for the labeling at the 5 prime end. The next is the methyl transferase. So the methyl DNA methyl transferase mediated modification of the bacterial DNA that restrict the endonuclease enzymatic cleavage of the host DNA, while the invading foreign DNA is open to enzymatic cleavage by the ligase enzyme. As we have already discussed that DNA ligases are used to ligate the two DNA, that is the gene of interest and the vector. The next is the terminal deoxynucleotide transferase. This enzyme catalyzes the incorporation of the single deoxynucleotides into the 3 prime OH terminus of single or double stranded DNA, and it is it has a various use in the 3 prime end labeling applications such as terminal DUTP, nick and labeling, also known as tunnel. The next is the Taq polymerase. Taq polymerase is a thermostable DNA polymerase 1, as it is the DNA polymerase, so it will help in the replication process, and it is isolate from the bacteria, that is Thymus aquaticus, which was isolated in 1976 and now known as Taq or Taq. The next is the cloning vector. So when we insert our gene of interest in the recombinant in the vector, and it will form the recombinant DNA molecule. Now how the cloning will occur? So for the cloning purpose, we will transfer it into the host organism. So what are the characteristics of the vector that we are used for the cloning purpose? So it must be small in size, so it can enter into the host organism. It must be self replicating inside host cell, so it should be independently replicating capability. The next is it must possess restriction site for restriction endonucleases enzyme, so we can uh, cut it and insert our gene of interest. Introduction of donor DNA fragment must not interfere with the replication property of the vector. So when we insert our gene of interest into it, it should not interfere with the replication of the vector. It must possess some marker genes such that it can be used for later identification of recombinant cell. So after the recombination takes place, then select the marker genes help us to identify the cells that contains this recombinant DNA molecule. And the last one is the multiple cloning sites. So in the case of multiple cloning site, we can use a number of restriction enzymes that can be used to clone the gene of interest. So now, as we know that plasmids and bacteriophages have ability to replicate the bacterial cells independent of the control of the chromosomal DNA, and the number of plasmid in each cells are varies, that may from it may contain from 15 to 100 copies of per cell, and it can go even higher. uh, Plasmids are non-chromosomal DNA. The replication process is independent of the chromosome. Many copies per are presenting in per cell. They are easy to isolate and they are easy to manipulate. That's why they can be used as a vector. The next is the bacteriophage. So as we know that when the virus infect the cell, it form a very high number of copies. So, if we link our gene of interest with bacteriophage or the plasmid DNA, then it will be very easy to clone our gene of interest. The next is the vector for cloning genes in plants and animals. So, as we have seen that the virus can be used that infect the bacteria. So, how we can use the DNA that can be used that are infecting the plants and animals. So, the pathogen like Acrobacterium tumefaciens, that are the pathogens for the dicot plant, they deliver their gene their gene, some part of their gene into the plant cell. That part is the tDNA, and that part is capable of induce tumor in the plant cell. And what we can do is we disarm the ability of the agrobacterium bacterium tumefacium to form the tumor, and we can use this pathogen as a vector for the plants. And the next is the retrovirus. We know that retrovirus are able to convert the normal cells into the cancerous cells. So again we can disarm the retrovirus. That means we remove we reduce the ability of the retrovirus to convert the normal cells into the cancerous cells, and then we can use them as the vector. So what retrovirus do is retrovirus enters the cell via the process of the endocytosis, then it uncoat itself and release its RNA. Okay, then with the help of the reverse transcription process, this viral RNA is converted into the double stranded DNA, and this double stranded DNA then enters our nucleus and incorporated in our our DNA chromosome. And with the help of the transcription process, again it will form its genetic material, and then it forms this RNA will produce proteins. It assembles itself, and with the help of the budding, new virions are formed. So, in in this case, what we will do is we, as it has a capability of incorporate itself in our DNA, then we will insert the gene of interest inside the inside it, and then it will be helpful for the cloning purpose. Now we will study about the plasmid, that is PBR322. So it can be isolate from E. coli. The size of this plasmid is 4361 base pair. The cloning limit is zero point 0.1 to 10 kb. That means the gene of interest having zero point 0.1 to 10 kb uh, can kilobase pairs that can be inserted inside the plasmid. The marker genes are used can be ampicillin and tetracycline resistance genes, and there are restriction site of for various restriction endonucleases like BAMH1, SAL1, PBU1, EcoR1, HIN3, etc. So, how the origin of PBR322 plasmid takes place, because it is an artificial plasmid. So, it is an artificial plasmid. So natural, how natural plasmids are helpful in the synthesis origin of the PBR322. So there are three major region of the PBR322. The first is the ampicillin resistance gene, the tetracycline resistance gene, and the third is the origin of replication. The ampicillin resistance gene is derived from the plasmid RSF2124, which is present in the E. coli. The tetracycline resistance gene is derived from PSC101, and the origin of replication is derived from PMB1, which is closely related to the col E1 that help in the production of the colicin. Now, in present day, the vectors are engineered in such a way so that they can help in easy linking of the foreign DNA with uh, the gene of with the vector, and it make the selection process easy for the selection of recombinants from non recombinants. So there are the features that are required to facilitate cloning into a vector. First is origin of replication, second is selectable marker, and third is cloning site. So origin of replication. So in a chromosome, there is a specific DNA sequence is present that is known as the origin of replication, that is responsible for initiation of the replication process. So there is a sequence where the replication starts, and when we link our gene of interest into with that particular region, then the replication of our gene of interest also takes place. And this sequence, the origin of replication, also controls the copy number of the linked DNA. So when we incorporate our gene of interest in the vector having origin of replication, piece of DNA will replicate and multiply itself in the host organism, and this process is known as cloning, or making multiple identical copies of any template DNA. The next is the selectable marker. So, selectable markers are used to identify and eliminate non transformant and selectively permit the growth of only transformant. Okay, so the transformation is a process through which a piece of DNA is introduced into the host or bacterium. In E. coli, the selectable markers used are ampicillin, chloramphenicol, tetracycline, or kanamycin. So here is an example that if there it is the master plate that carry the E. coli, and this E. coli contain the plasmid vector, and the vector is okay. So this is the vector contain the PBR322. We incubate this plasmid with the E. coli. So what basically happens is this when we cut this DNA here with the restriction enzyme, and we incubate it with our gene of interest, then there are two possibilities that if it may enter uh, this gene of interest may enter into the vector, incorporate into the vector, or it may not. That means it will religate. Okay, so when we incubate the in recombinant DNA with the E. coli, then there are three possibilities that the uh, plasmid will not go inside the host organism. That means inside the E. coli. Then there are non transformed cells, and if the religated uh, plasmid enter inside the E. coli, then the cells are transformed, but the plasmid enter into it is not recombinant. So the cells are non-recombinant have non-recombinant vector. But when the cells have this type of plasmid, that means the cells contain the gene of the plasmid has gene of interest, then the cells are called as transformed and recombinant vector. Okay, so first, these we don't know that which colony have which colony is not transformed or which colony is not transformed. So what we will do is, for the selection of the required colony, first we will grow these cells on the ampicillin containing medium. So if the cells do not contain any plasmid, as E. coli do not have any antibiotic resistance gene, so the cells of the E. coli that do not have the plasmid that do not have that plasmid into it, they will not grow on ampicillin containing medium. That means non transformed cell will not grow in the ampicillin containing medium. Only transformed cell will grow in the ampicillin containing medium. Then we will uh, we will transfer this colony on the tetracycline containing medium. Okay, so with the help of the replica plating. So in this case, what will happen is, if the plasmid containing the gene of interest, so the plasmid in which gene of interest is present, where the tetracycline resistance is now inactivated because the gene of interest is uh, enter inside the tetracycline resistance gene, that is known as the inactivation of the tetracycline resistance gene because of the insertion of our gene of interest. So here the tetracycline resistance property of the plasmid will gone, and here in this case the plasmid is now tetracycline resistant. So replica plating is a type of the plating in which is helpful in the selection process. So now these yellow colonies are the transformed recombinant colonies. So these colonies are plated on the tetracycline tetracycline containing uh, medium plate. With the help of the velvet, we transfer this colony on this plate. So as we know that both these colonies have the plasmid that has our gene of interest. So they are now sensitive to tetracycline, and they will not grow in this plate. Okay, so we will replicate it into the with the help of the replica plating, with the help of the velvet. So now we can choose that these two colonies are our desired colonies from the other colonies. Now, as we know that the inactivation of the antibiotics is a very cumbersome process, because it requires simultaneous plating on plates having different antibiotics. So there is an alternative selectable markers that are used for the differentiation between the recombinant and from the non-recombinants on the basis of the chromogenic substrate. So one of the process that include the uh, cloning the gene like Z that codes for beta galactosidase. This is again based on the insertional inactivation of the gene. Okay, so the basic reaction is The when the beta galactoside these enzymes is expressed by the LEGZ gene when the inducer and the substrate is there. Inducer is the isopropyl beta D1 thiogalactopyranoside. When it is present, it will express beta galactosidase, and when the substrate is present, that is XGAL, it will be uh, it will be converted into 5 bromo 4 chloro 3 hydroxy indole, and with galactose it will form a blue color compound. That means if beta galactoside enzyme is active, blue colony will be formed. Okay, so now if this is the plasmid vector and this is the foreign DNA, so there are three possibilities. First is the within insert is present within the leg Z, insert is outside the leg Z. Insert is with, this is the insert red color and blue is the leg Z gene, and there is no insertion of the gene of interest in the vector. So what will happen is, if the our insert is present within the leg Z, that means there is the insertional inactivation of the leg Z gene. And as we know that if the beta galactosidase is not expressed, blue colony will not form and white colonies will form. And in other cases, when the insert is outside the leg Z, or if there is no insertion, then the leg Z will express and then it will form blue colonies. So the first one, the white colonies are the here recombinant colonies. Now the cloning site. So the vector needs to have very few and preferably single cloning site. Why? Because in this case, as in we can see in PBR322, if there are more than BAM H1 sites are present in this vector, so it will be difficult to clone our gene of to insert our gene of interest in the BAM H1 site, because it has two BAM H1 sites, so we will not know that at what position our gene of interest is inserted. Okay, now, so the presence of more than one recognition site within the vector will generate several fragments and it will complicate the gene cloning. The ligation of the foreign DNA or alien DNA is carried out at a restriction site present in one of the two antibiotic resistance gene. For example, if the foreign DNA is inserted in the tetracycline resistance gene, then the resistance due to the insertion of foreign DNA, but can be selected out from non-recombinant ones by plating the transformant on tetracycline containing medium. The next is the competent host for transformation with recombinant DNA. So, as we know that DNA is a hydrophilic molecule and it cannot pass through the cell membrane, that's why the cells, that means the host cells, are competent cells. So there are some methods that are uh, help that helps us to make the cells competent. So one is the chemical method, in which the specific concentration of divalent cation like calcium is provided to the bacteria, so that the cell wall of the bacteria will be porous, and now the DNA can enter into the cells. And the other method is the heat shock method, which is also known as chemical transformation, in which the host organism is incubated with the plasmid, and the heat shock is given, which is 42 degrees centigrade for 30 seconds. And in this case, the cell wall is porous, and plasmid easily now get into the bacteria. And after some time, with the recovery, now the plasmid is inside the cell. The other method is the electroporation method. In the electroporation method, the host cells are again uh, incubated with the plasmid. Electric shock is given, which is 15 kilovolt per centimeter for 5 microseconds pulses. Again, the it makes the cell wall porous, and with the recovery the plasmid gets into the cell. And there are other method also, mechanical method like the micro injection method, in which the DNA is directly injected into the nucleus of an animal cells. In case of animal cells, there the another method in which the cells are bombarded with the uh, with the gene of interest. Here the gene of interest are covered on the gold particles, and with force, this is the plastic disc, and the gene of interest are present on the gold part. The recombinant DNA are present on the gold particle, and these gold particles are then bombarded on the target plant cell, and now the plasmids are present inside the cells. And the last method is used is the disarm pathogen vectors, as we have discussed that in case of plants we use agrobacterium tumefacium, and in case of animals, retroviruses can be used for the transfer of the recombinant DNA into the host. So this is it for this session, and we'll continue this chapter in the next part of the next part next part. And thank you so much for listening.
गुड। तो तुमने तो मेरा टॉपिक सारा पढ़ा दिया।
हाँ, पर सर इसमें ये था सब कुछ।